Okay, so buenos dias, everybody. Thank you for the opportunity to discuss this topic and to put together this plenary session. I have no conflicts of interest to report. I just want to go right to what our session will be. We have a very integrated, interesting format. We're going to have really 6 discussions or topics. I'm going to give an introduction about the HPV genome. Our keynote speaker, I'm very happy that can be here with us, Dr. Rob DeSalle, and I'll introduce everybody within this introduction. And the makeup of our session will be to talk about HPV genomics and its applications. We'll talk about some phylogenetic inferences from the genomes. We'll talk about new methods of actually determining genome sequence. We'll talk about bioinformatics, applications for the genome. And finally, I'll give you an update on the use of genomics for nomenclature as is the official representative of the ICTV. So first, let me start by what is papillomavirus genomics, or what are genomics in general? And I've given 2 definitions. Actually, it's very simple. The study of genomics is where the centerpiece of study is the genome, not necessarily the virus capsid, not the proteins necessarily, but in fact, the center of study is the genome. And there's 2 different definitions that I've written. One is from Wikipedia, and one is from a recent review by Francis Collins and Alan Guttmacher from the NIH. And they expand the idea of genomics in the study of genomics, but we're primarily concerned with the genome. So as far as HPV genomics, what does this really entail? Well, it has to deal with viral heterogeneity. What is the extent of the different viruses that exist on the planet today, their description and how they got there? I think for most of the people in the room, it probably deals with the genotype-phenotype associations, in that specific genotypes are associated with cancer and are a public health interest and result in one of the more common forms of cancer in women and now other areas of the body. Also evolution, the evolution of these genomes, how did they come to exist today as we can measure them, since it's very difficult to measure them thousands of years in the past. They also can give us clues to the evolution of the host in some very unique ways as an independent marker. Those studying structure function can use genomic comparisons to look at a function. But I want to remind everybody that really within the genome there's coded information. We usually think of the nucleotide sequence, the open reading frames, but there's lots of other information that's there that we haven't begun to appreciate. There's splice sites, there's DNA protein binding sites, there's a whole CPG code that is just coming of interest and of aware not only in the papillomavirus genome, but in the human genome in general. And I'm sure there's lots of other codes that have evolved over the millions of years that these viruses have survived. And lastly, we use the genome for nomenclature of isolates. So let me just give you kind of my take, one way of looking at the genomics of papillomavirus as a medical geneticist, and that's everybody's aware of the inherited risk for particularly a woman that has a BRCA1 or 2 mutation. This was discovered through linkage analysis and then sequencing the gene and its risk for breast cancer. Well, in the papillomavirus field, we don't have linkage analysis. What we have is association. And we've been able to show that persistent HPV-16 can some ways be equivocated to the inheritance at the somatic cell level of a viral genome, which has enormous cancer risks. This could be one of the most carcinogenic activities that we're aware of. However, it's not just persistence that's the story. We know that persistence with HPV-61, in fact, has no reported abnormalities. And the focus of genomics is trying to understand these differences. And this is now pointed out in this slide, where we have 2 well-known viruses their open reading frame structure is essentially identical. HPV-16 on the left and HPV-31 on the right, but their biologies are uniquely different. This data below is from a recent paper by Gary Clifford's group, where in normal individuals they found about 20% of the women had HPV-16, where for HPV-31 they found about approximately 8% prevalence in normal women with normal cytology. In SIN3, the prevalence of HPV-16 goes way up to 58%. In HPV-31, it goes modestly up. And if you look at the cancer, HPV-16, as we all are well aware, is responsible for 63% of cervical cancer worldwide, whereas HPV-31 is only responsible for 4%. And if you look at the ratios of cancer to normal prevalence rates, you see that HPV-16 is 3, where HPV-31 is 0.5. If you look more importantly at SIN3 to ICC, it's about 1-to-1 ratio, where HPV-31 is 0.3. So the question is, what are the underlying genetic determinants of these biological manifestations? It also raises the point of how good a marker SIN3 is for cervical cancer. For 16, it appears to be an exceptionally good marker and genetically determined, but for HPV-31, you see that invasive cancer is far less common than SIN3. This story is even more prominent for HPV-51, which is a common cause of SIN3, but really found in cervical cancer. And the study of genomics, the genotype-phenotype correlations, is really trying to understand what are the actual genetic basis of these differences. So just to kind of summarize the genetic determinants of HPV pathogenicity, so by empirical means, we've identified 12 oncogenic HPV types. We use association as our main means of identification based on population-based studies. We have effective vaccines developed against these oncogenic types, and we're entering now an era of HPV genomics where the genomics will be testing for cervix cancer prevention. This is all a use of our understanding of HPV genomics. Although we don't think about it, we accept just the risk of HPV-16. Those of us that are interested in the genomic side are really trying to understand what are the specific determinants of the pathogenicity. Now, I'm going to go through all our speakers, and then I will call them up without much of an introduction. So our keynote speaker today, who I'm very delighted is able to join us. He suffered terribly from the recent hurricane in New York City. Puerto Rico is not the only place that has hurricanes. We had a devastating hurricane in New York City. If you want to talk to Rob DeSalle afterwards, he can talk about 11 feet of water in his basement. So we're very grateful that he was able to make it. The title of his presentation is going to be The Human Genome and Human History. He's also going to tell us about the Tree of Life. Rob is a curator at the American Museum of Natural History, where he was the lead curator for the Genomics Revolution exhibit and the curator of the Hall of Human Origins if you've been in New York City and visited the American Museum of Natural History. Curator is an interesting role that is unique to natural history museums. He's also a member of the Sackler Institute of Comparative Genomics. He's a distinguished professor in residence at NYU, and he holds many other important titles. He's the recent co-author of Human Origins, What Our Bones and Genomes Tell Us About Ourselves. That was co-written with Ian Tattersall. So he is a real expert on genome analysis and human origins. Our second speaker will be Dr. Bravo from Spain. The topic of his talk will be on the phylogenomics and how it reveals different modes of evolution for different genes. He's now the head of infections and cancer laboratory at the Catalan Institute of Oncology in Spain. And he trained with some well-known persons in the HPV field, Angel Alonso and Lutz Gissman, where he studied the biological and evolutionary functions of E5. And he then went to Montreal to learn phylogenetics, and he's been interested in the evolution and selective pressures of the HPV genomes for a number of years. Following Dr. Bravo will be Dr. Kukimoto. He's the Chief Pathogens Genomics Center and the National Institute of Infectious Diseases in Japan. His topic will be the use of new technology, next-gen sequencing, and specific polymerases for the identification of HPV-16 full genome sequencing. More and more we're going to hear and learn about the introduction of new technologies into genome analysis, both for the basic science use and for diagnostics. He was a visiting professor or scientist at the Madame Curie Research Institute in the UK, and we see a nice picture of him with his family. Okay. Following his talk, we're going to move into bioinformatics, where Conrad van Doorslayer will talk about PAVE, or the Papillomavirus episteme. This is a bioinformatics software tool which is available online through the NIAID. Vani is in the lab of Alison McBride, and he's contributed significantly to the development of PAVE, and we'll talk about some of its features as it'll help us all in the community understand and decipher HPV genomes as we move more towards a sequence-based detection of HPV. And then I will end up the platform with a discussion of the new things in papillomavirus nomenclature from the ICTV. I'm the chair of the papillomavirus study group, and I'll tell you some new things that are going on and some other things that will address the community. This is community service, so we keep everything straight.